So, hello everybody. Thank you for being here. I'm Shilpa Gadgil from Enzene. So, let me start by telling you that I'm going to tell you a story. And this story is about an idea that started back in India almost a decade ago. And over time, it has turned into something that we genuinely believe that industry needs as of today. Okay? And this idea is what we refer to as EnzeneX. Essentially, what we have done is it's an attempt to re-engineer how the world makes biologics. Okay? So that is what I'm going to talk about. It is going to be a journey. So I'm going to talk about where we come from, where we are currently, and then where we are heading. So past, present, and future of EnzeneX journey that has become an industry imperative today. So when we started, we looked at, we stopped dead in our tracks, in fact, because we looked at the cost of gold per gram, which is one of the most precious metals. And then we looked at cost of monoclonal antibody per gram, okay? And after 10 years, the picture still remains the same. So cost of gold per gram today is about $110 per gram, and cost of MAB per gram ranges anywhere between $180 to $500 per gram. The unfortunate thing about this is that as an industry, we have learned to accept this fact that life-saving medicines are costlier than gold, and there is acknowledgement, but very little has been done to reduce the costs so that the medicines become affordable. So our initial goal was to democratize access to life-saving biologics for patients in India and emerging markets. This is especially more important because the patients in India as well as emerging markets pay out of their pockets. So affordability is critical. So when you ask about affordability, how you bring that into picture in manufacturing, you face some hard truths, right? The fed batch, you can get, by modifying whatever you have already, the fed batch optimization, you can scale up or you can have media. You can get, you can get incremental improvements, a 10% gain here, a 20% gain there, but the math doesn't quite close the gap. So what we have to do is a change in order of magnitude is required. And how do you get that? That you would get by reduction in capital costs, operation costs, and as well as reduction in footprint. So when you ask the question, what do we have to change to be able to get that incremental or huge change, step change to get the affordability in picture, right? So the answer remains, we pretty much have to change everything. So then the question was, we wanted to re-engineer the process, from the fundamental way how we make the biologics. Re-engineer the process. You can make a bigger and better plant, but that's not what we wanted to do. We wanted to re-engineer the process, and that we would have to make the case for continuous manufacturing. So between 2015 and 2019, that is what we did. The core problem remained the same. The conventional biomanufacturing, fed batch, it's all the unit operations are standalone. You would have whole tanks. So essentially, you're running the process in one unit operation, then you collect, then you hold, then you transfer it to the next one. Okay? So it is a chain of discrete stop, start, transfer unit operations, right? What happens is that, you lose time, there is loss of productivity, and then there is a lot of capex, which adds into your COGS. So what we thought was, why don't we do process intensification, which is done already. It is nothing new. So for upstream, what we did was, we did process intensification by using perfusion bioreactor along with ATF, which is the cell retention device, where the media will flow continuously with the product, and the volumetric productivity goes through the roof, almost 10 to 30-fold increase compared to the fed batch. Another thing that we did was, we also did intensification in downstream. So what we did was we used multi-column chromatography systems. For protein A, we have two-column parallel mode systems. Then we also connected the entire Chrome systems, from Chrome 1, Chrome 2, and Chrome 3. So everything was running together. And then you also end up with smaller resin volumes. All of these changes add up in reduction of COGS. And then the final leap, or the final most important thing that we did, was we connected the entire upstream with entire downstream up to Chrome 3. A lot of people keep talking about having a continuous manufacturing. You can have just upstream as perfusion, that is continuous manufacturing. You can have downstream that is all connected, that also people refer to as continuous manufacturing. Or you can have upstream that is connected with your capture chromatography, and that people also refer as continuous chromatography. What separates EnzeneX, which is the FCCM, or fully connected continuous manufacturing, is rather than having the entire connection, there are no whole tanks. So what is missing is the whole tanks, okay? And then we have connected everything from upstream bioreactor, connected with your ATF, which is the cell retention device, then Pro-A, which is the capture chromatography, virus inactivation, anion and cation exchange in tandem mode, and then there is a whole tank. So in the entire processing from upstream to Chrome 3, there are no whole tanks. And that reduces your footprint, your capex, humongously, okay? The nanofiltration, UFDF steps are standalone unit operations. So EnzeneX 1.0, so what you just saw was designed, we designed it, but we also proved it. And what you can see here is the actual picture of the GMP suite that we have back in India. It's a complete connected continuous line in 1,500 square feet. It is GMP proven. We have scales from 50 liters to 500 liters as part of our EnzeneX 1.0. The productivity I want to focus on, it goes at least 10-fold high. The COGS are minimum 50% reduction that we see compared to fed batch. Again, I want to stress on the low footprint, which again results into lower COGS and lower cost per gram of your antibody or whatever molecule you are making. The capex that goes into it, it's around $80 to $90 million compared to 300 to almost $500 million that you would require for a traditional fed batch plant, So essentially, because our suite is very small, it can sit somewhere in the corner of the entire big fed batch plant. And therefore, what happens is the COGS, or cost of goods, goes down from $60 to $100, okay? Now, when I started my talk, I was talking about the COGS per gram of monoclonal antibody range anywhere between $180 to $500. With EnzeneX 1.0, the COGS are down to $60 to $100 per gram. So we normally get a question that, EnzeneX 1.0, that's fine, it works. Does it work in GMP? Is it a pilot plant? So I want to stress that point. It is not a pilot. It is not clinical manufacturing. It is definitely not a demo. It is a working GMP plant where we have launched several molecules. So this platform, again, it is not a pilot. It is a commercial reality. We have launched 5 biosimilar products using this platform in India. We are very routinely manufacturing 50 kegs of 50 kegs of product in a 1,000-liter FCCM batch, so the scale-up also has happened from 500 liters to 1,000 liters. And then we have scaled out. We have a US site that is fully functional at Hopewell, New Jersey, where we are able to manufacture using either FCCM or fed batch. And I want to take a moment, and we are very proud that we were awarded the CPHI 2025 Manufacturing Excellence Award last year for this EnzeneX FCCM platform. So EnzeneX 1.0 established the FCCM platform. It works. And EnzeneX 2.0 is where the economics have been redefined. So what I mean by that is that this is the next version of EnzeneX 1.0, where the COGS were reduced even further. The driver for this COGS reduction was higher titer cell lines. Okay? So in 2-liter perfusion bioreactor in the FCCM, we were able to get 135 grams of protein. We have scaled it up to 1 kl from 2 liters. And the COGS went down from $90 per gram to $40 per gram. And that is where we are currently. Another question that we get, is it a one-time deal, one-time wonder? Does it only work for MABs? So the answer is no. It works very, very consistently. Like I said earlier, we have launched already 5 biosimilar MABs. These are new 3 biosimilar MABs that I'm telling. And you can look at the growth profiles here and also the productivity per day. Very, very consistent across molecules. So the platform works really well. It's very consistent. So the platform not only works for MABs, we have also used FCCM to work on bispecific monoclonal antibodies, bispecific, multispecific, non-MABs. This is very, very, very important considering the new generation of drugs that are coming up, which are very low dose. So it works beautifully for that because they are highly fragile. It works for fusion proteins. And then also we have done several proof of concept studies where we have converted the existing fed batch process into FCCM. This is just one of the examples of a bispecific non-MAB. This is human engineered, highly fragile molecule, and I will not spend too much time on this, but what I want you to focus on is the silver stain gel where we got single band with the FCCM, as opposed to or in comparison to a fed batch, which looks like a ladder in Kumasi. So the material quality remains really good in FCCM. What was the key impact of this study? So we got 27 grams of protein in 15 days using a 20-liter bioreactor compared to 3 grams in 50-liter fed batch. Okay, this is a low-dose product, so we have been able to get more than 500,000 doses using this drug substance. Okay, and with this, the client was able to carry out their phase 1 and phase 2, and still the material is left over. The entire part we were able to finish in 6 months, the entire development and then the clinical trial manufacturing. One of the coolest things about this platform is that there are 3 ways to scale this platform, okay? You can scale up, so essentially a bigger bioreactor just like your fed batch. We have established the scale up from 2 liter to 1,000 liter. So this is not done in theory, this is real example. You can scale on, so you're essentially taking the same bioreactor, you're just extending the duration. This also has been done in practical, so from day 20 to day 36. We have done this in our commercial products as well. One of the neat things about it is you can scale out very easily. Because the footprint is very small, it is very easy to add multiple identical suites so that you can meet the commercial demands whenever required. So just like you can have a suite next door to meet your demands, you can scale out geographically. And what we have done is India, Pune site is our hub. And we have scaled out to US, New Jersey, Hopewell, New Jersey. That's our plant, which is fully functional. FCCM batches are being taken there as of today. The coolest thing is, you can take this same platform, you can take the entire platform and bring it to your own territory, right? So it can go wherever the patients are. So this is a manufacturing platform that travels where the patients are. And this is called as NEX. So this brings us to the future of EnzeneX. So I'm going to talk a little bit about our next platform, and I just want to make an announcement. We are inaugurating or launching our next program at 5 o'clock at booth 5700 at our Enzene booth. So please come and attend the inauguration. So next, scaling out to your own territory. So this is a hub and spoke model that essentially transfers the whole capability. The operating word here is the whole capability, right? So what we are doing is that the India site will transfer the entire capability to your territory. So the spoke site essentially gets the tech transfer of the entire EnzeneX platform. We will be able to do the remote batch monitoring and real-time troubleshooting. We will provide all the process and analytical expertise that are required. We also have our own biosimilar pipeline and clinical packages, which we will give it to you. Last but not the least, the workforce that is required to run the process, we will train it through our Enzene Academy, which is called as Sparks. So essentially, you will be able to manufacture your products, that you can take from us, or your own products that you develop eventually. And you will not have to worry about any pandemics or tariffs or supply chain issues, right? You will have your local manufacturing. Another thing that we are doing currently is integration of advanced PAT. That should be available by the end of 2026. Unified data flows. And essentially what is going to happen once we incorporate PAT is that it will take one more layer of QC testing away, and again, it will have an impact on reduction of COGS. So the most important thing that will happen with this is, from reactive monitoring, you will go to proactive, intelligent process control. So where do we want to go from here? So our vision is that we want to be a Tesla of biomanufacturing. The process has to be auto-driven, biologics production. It has to be predictable, efficient, and self-optimizing operations. So what do we want to do? We already have the foundation in place, which is EnzeneX 1.0, that is proven. EnzeneX 2.0 is where we are currently. The scale-up already to 1,000 liter has happened. We already have high-titer cell lines. Moving forward, next year we will have the advanced PAT already included as part of our EnzeneX 3.0. Again, next will be also part of that. We will also go for media innovation. We will have in-house media, which will again drive the costs down significantly. And then eventually we'll have AI ML layer included. The goalpost that we are looking at is that the COGS should be $10 per gram for the antibody production. So just to summarize, what do we have? We have the process that is fully proven, that is the foundation, the fully connected continuous manufacturing. The proof is in the pudding. Commercial portfolio, we already have 5 biosimilars. 5 biosimilars are already marketed in India. Several INDs supported using this FCCM platform. The scale, 3-way scale, up, on, and out. And you can also have the next that I was talking about, geographic scale out. And then future, we will have PAT, next, and autonomous operations. So essentially, the innovation that becomes industry imperative, the EnzeneX journey continues. And the next journey, the next chapter, the next territory could be yours. So, I'm happy to answer any questions. With that, I'd like to stop. Thank you.